The Midterm project by Barbara
and Layla.
The pH effect on enzyme lab. In this lab, we are simulating the effects of rattlesnake venom, which contains enzymes that break down certain molecules. We will examine how enzyme activity changes when exposed to different pH levels, and measure how long it takes for the venom to deactivate its target under these conditions. The procedure. We started our lab by turning on one of the hot plates and placing a big beaker filled with water on it. While waiting for the water to fully boil, we measure the amount of enzyme and substrate. To test the effect of pH, we prepared 3 test tubes. The first one was filled with water, which is neutral pH, the second one with vinegar, acidic pH, and the third one with bleach, basic pH. In each test tube, we added the combined mixture of enzyme and substrate. We shook the tubes gently to mix them thoroughly, and waited for 5 minutes to let the enzyme and substrate interact. Once the 5 minutes passed, we carefully placed all 3 test tubes into the beaker of fully boiled water on the hot plate. We started a timer and observed the tubes, recording how long it took for the solution in each tube to change color.
Our research question is, how does pH affect the activity of rattlesnake venom as an enzyme to deactivate its target? Background information. Enzymes are biological catalysts that speed up chemical reactions. They work by lowering the activation energy required for a reaction to take place. Enzyme activity is highly influenced by pH. Each enzyme has an optimal pH at which it functions more effectively. At pH levels below or above this optimal range, the enzyme's structure and active site can be altered, leading to a reduced activity or complete loss of function, which is known as denaturation. Our hypothesis is, if the pH is closer to the enzyme's optimal range, then the time it takes for the rattlesnake venom to deactivate its target will decrease. However, at extreme pH levels, the enzyme's activity will decrease or stop due to denaturation or disruption of its active site. Our control group is a sample where the enzyme activity is tested at a neutral pH of 7. The experimental groups are the samples where the enzyme activity is tested at various pH levels. Our controlled variables are the concentration of the enzyme, temperature, mass of the target substrate, and reaction time for measurement and measurement method.
Our independent variable is the pH of the solution in which the enzyme activity is being tested, and the dependent variable is the time taken for the enzyme to deactivate its target or the level of enzyme activity measured over time. This is our data.
In conclusion, the results show that the pH significantly affects enzyme activity. The water, being neutral pH of 7, allowed the enzyme to function most efficiently, resulting in the fastest color change. The base with the higher pH slowed the reaction, indicating reduced enzyme efficiency. The vinegar, which is acidic, prevented the color change entirely, suggesting that the enzyme was denatured or inactive at low pH levels. This highlights that enzymes have an optimal pH range where they work best, and extreme pH levels can inhibit or stop their activity.
Macromolecules. Macromolecules are classified into carbohydrates, lipids, proteins, and nucleic acids. Macromolecules consist of monomers. A monomer is a building block. It usually refers to the same type of block that makes up something larger. Carbohydrates. The monomer of a carbohydrate is the monosaccharide, for example, glucose. If you join many monosaccharides together, it creates a polymer. Carbohydrates provide energy for cellular activities. For example, glucose is broken down during cellular respiration to produce ATP. Lipids. Most lipids have building blocks of glycerol and fatty acids. Lipids form the structural basis of cell membranes, such as phospholipids, and store long-term energy. Protein. A monomer of a protein is an amino acid. Protein acts as enzymes to catalyze reactions, form structural components such as keratin in hair, and transport molecules such as hemoglobin for oxygen. Nucleic acids. Nucleic acids have a monomer called a nucleotide. Nucleic acids store and transmit genetic information, DNA, and assist in protein synthesis, RNA.
Protein folding.
After translation at the ribosome, proteins undergo folding into specific 3-dimensional structures essential for their function. The folding process occurs in 4 levels. Primary structure. The first level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. They are held together by peptide bonds. The primary structure determines the order of amino acids, and the properties of these amino acids guide the folding process. Secondary structure. Local folding into structures such as alpha helices and beta plated sheets, stabilized by hydrogen bonds. Secondary structures are the result of interactions between the backbone of the polypeptide chain. Tertiary structure. The overall three-dimensional shape of a single polypeptide chain stabilized by various bonds and interactions. The tertiary structure is determined by interactions among the side chains or groups of amino acids, creating a functional, stable protein. Quaternary structure. The arrangement of multiple polypeptide chains into a single functional protein. Examples include hemoglobin. Quaternary structure results from interactions between polypeptide subunits to form a larger functional complex. Did you know? Misfolded proteins can cause diseases like Alzheimer's and Parkinson's by forming harmful clumps in cells. Understanding protein folding helps scientists design therapies to prevent or treat these conditions.
Gram-positive and gram-negative. Bacteria can be classified based on the reaction to the gram stain, which identifies them as gram-positive or gram-negative based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall which consists up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall consisting of a single layer of peptidoglycan. This layer is between two lipid bilayer membranes called diterms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then a counterstain is added. Gram-positive bacteria will retain the crystal violet dye and remain purple, while the gram-negative bacteria will be stained pink. The reason for this staining differences is due to differences in cell wall structure. The gram stain detects peptidoglycan, and since gram-positive bacteria have thick multi-layered peptidoglycan layer, they retain the crystal violet dye. Gram-negative bacteria do not retain the dye because they have an outer membrane getting in the way of the dye, and they lack peptidoglycan to retain the stain.
How antibiotics work. Pathogenic bacteria in the body cause infections, which can be treated by antibiotics. Antibiotics can be bacteriostatic or bactericidal. Bacteriostatic antibiotics slow the growth of bacteria by interfering with the processes the bacteria need to multiply. These processes include DNA replication, metabolism such as enzyme activity, and protein production. Bactericidal antibiotics kill the bacteria, for example, by preventing the bacteria from making a cell wall. Penicillins are bactericidal. Beta-lactam antibiotics, such as penicillin, target the enzyme transpeptidase, which is essential for bacterial cell wall synthesis. By binding to transpeptidase, beta-lactam antibiotics prevent the cross-linking of the peptidoglycan layer in the bacterial cell wall, causing the bacteria to weaken and rupture.
Antibiotic resistance. Antibiotic resistance occurs when an infection responds poorly to an antibiotic that once could treat it successfully. It is the bacteria that has become resistant to the antibiotic, not the patient. Mechanisms. This happens because the bacteria acquire a mutation that gives them a new protein as a tool to fight the antibiotic. Some of these different mechanisms are: it can prevent antibiotic from entering the bacterial cell. It can pump the antibiotic out of the cell. It can destroy the antibiotic by enzymatic reaction. It can modify the antibiotic's target so it no longer binds to the drug. It can give the cell a way to bypass the antibiotic's target, making the drug irrelevant. How antibiotic resistance emerges. In situation when a new mutation emerges and makes the bacteria resistant to a certain antibiotic, in the absence of the antibiotic, such mutation offers no advantage, and because mutations come with slower growth, the mutation would be diluted and eventually disappear in the following generations. On the other hand, in the presence of the antibiotic, only bacteria that carry such mutation would survive, and they would soon take over the whole bacterial population. The use of antibiotic is the factor that drives the selection of antibiotic-resistant organisms. How antibiotic resistance spreads. Mutations that confer antibiotic resistance can be transmitted not only vertically from parent cells to offspring, but also horizontally, from one bacterial cell to another using mobile genetics elements such as plasmids or bacteriophages. This means a bacterial strain can share their antibiotic resistance with other bacterial strains and even with distantly related bacterial species. These antibiotic-resistant bacteria can infect humans, animals, and spread between them through food and the environment. Antibiotic resistance is one of the biggest global health concerns. Infections by antibiotic-resistant bacteria are much harder, sometimes impossible to treat. Some bacteria called superbugs are resistant to most antibiotics and are especially kill. Treatments for infections caused by such bacteria are costly and toxic to the patients. To help control spread of antibiotic resistance, antibiotics must be taken correctly only when prescribed by a healthcare professional who should do so only when antibiotics are needed. Antibiotics should not be used to promote growth or prevent diseases in healthy animals. Measures that help prevent infections also help reduce antibiotic overuse.
Medical intervention. The medication name is called resistance 1 and the class of macromolecule is a lipid. How it works. Resistance 1 is a lipid-based drug designed to insert itself into the bacterial outer membrane. It forms a physical barrier that blocks antibiotic resistance pumps from ejecting antibiotics out of the bacterial cell. This allows antibiotics to accumulate inside the bacteria and kill it effectively. How it reduces antibiotic resistance. At the cellular level, it targets efflux pumps on the bacterial membrane, preventing the pumps from expelling antibiotics. This increases the intracellular concentration of antibiotics, allowing them to attack targets like the cell wall or protein synthesis machinery. At the population level, by limiting bacterial survival, fewer resistant bacteria remain to reproduce or share resistance genes among others. This is a simple bacterial cell with an efflux pump actively ejecting antibiotics. Resistance 1 inserts into the pump and blocks the antibiotics. The antibiotics stay inside the cell and accumulate, turning the cell red. The bacterial cell turns gray, indicating that it has been killed.